guys, Rob from Royal Balls. This is going to be the second part of my genetics series, and today we're going to deal with some terminology that we need to know before we go on to more advanced stuff. So let me just recap what we learned in the first video. We dealt with a single codominant or incomplete dominant gene. We looked at the pastel gene, and this is the pastel animal. And if you remember from the first video, this animal carries a single copy of the pastel gene. So here in my genetic code we have the DNA strand from the male, the DNA strand from the female, and we learnt that this particular animal with one copy of the genetic material, when that is passed on to the offspring, the genetic code is split in half, and only one half of the genetic material carries the gene. And if we pair that to just a normal ball python on the other side, you will remember that when you pair this half of the male to that half of the female, or that half of the female, the gene gets passed on. One copy from the male, no copy from the female. And when we pair that part of the male to this half of the female or that half of the female, none of it gets passed on. So in this particular case, using my Punnett square, you don't really need to know that, but just acknowledging the guy that invented it, we see that in the case of a single gene with a single copy, the gene is passed on 50% of the time. The two offspring here will be pastels, visible pastels. The two offspring here will be visible normals. Okay, so that's where we'd got with a single incomplete dominant or codominant gene. So I'm going to look now at what we mean by dominant, codominant and recessive genes. They're three terms that we need to understand before we move forward. So let's deal with the dominant gene first. An example of a dominant gene would be a spider. This is a spider ball python. This carries a single copy of the spider gene. So just like the dominant or the codominant, this animal has a single copy. Now the dominant gene is so dominant that one copy totally changes the appearance of the snake. If you remember from the incomplete dominant, you can have a superform. So one copy changes the visual appearance slightly. The superform gives you the full expression of the incomplete dominant gene. In this particular case, the full expression of the gene is exhibited by just one single copy. And adding a second copy of the gene, so powerful is this gene, adding a second copy is actually lethal to the animal. So examples of dominant genes would be spider in this case, champagne, warmer, and a number of other genes. But generally speaking, there is no superform. If you attempt to make the superform, the snakes generally die in the egg. So one copy is present in that animal. And just as in the single copy of pastel, the outcome is exactly the same for that animal. If you breed a spider to a normal, the spider carries one copy of the gene, so 50% of the babies will carry the spider gene and 50% will be normals. Now let's just see what will happen if we attempt to breed a spider to a spider. And I've said that that particular outcome is lethal. And I know that some people have mentioned that they have bred a dominant gene to a dominant gene and some of the babies survived, so how can it be lethal? Well, let's just have a look at what the potential outcomes are when we breed a spider to a spider. So the male and the female both carry a single copy of the dominant gene. They split their genetic material in half. So what happens if we take this half of the male, we pair to this half of the female in the offspring. So in this square we get two copies of the gene. This half of the male to this half of the female, only one copy of the gene. We take the other half of the male's genetic material, which isn't carrying the copy of the spider gene, we pair it to this, which gives us one copy of the gene from the female, but no copies from the male, or we pair it to this half of the female genetic material, and in this particular case, there are no copies of the gene. So we have 4 potential outcomes. This square has no copies of the gene, so is a normal. These 2 squares here have a single copy of the gene, so they will both be visual spiders. This square is the only square that carries 2 copies of the spider gene, and this is the lethal combination. So yes, if you pair spider to spider, some of the babies will survive because they're only carrying a single copy of the gene. 1 in 4 of the babies will be the superform, and that is lethal. So that is why we don't like to pair dominant gene to dominant gene. We don't pair spider to spider because 1 in 4 of the babies will be dead in the egg. Okay, so that covers a dominant gene, a gene that is so powerful that it totally changes the appearance of the snake in 1 copy of the gene. We've already dealt with pastel, which is a co-dominant or incomplete gene, and we know that that has a visual form with 1 copy and it has the superform with 2 copies. So that covers incomplete dominant or co-dominant. What do we mean by a recessive gene? Okay, a recessive gene would be something like pied or clown, and we'll use pied as an example. This happens to be a pastel pied, so it has, because it's a visual pied, it is carrying 2 copies of the pied gene. A recessive gene does not show up in the offspring if the offspring only have 1 copy. The way to visualize this, I think, is from your school days in basic biology, when you looked at human biology. Blue eyes. Blue eyes are recessive, which means that if one of your parents has blue eyes and one of your parents has any other eye color, you will not get blue eyes. All the children from that marriage will not have blue eyes. In order to get blue eyes, both parents must have blue eyes, and they pass on 1 copy of the gene, 1 from the male, 1 from the female, so the offspring are all carrying 2 copies of that recessive gene, so the babies can have blue eyes. So this is a pied. Clowns, albinos, lavender albinos, a whole host of recessive genes that require 2 copies of the gene to have a visual expression. So let me just put the snake back and we'll have a look at this. So automatically, if we have a visual pied, we know it's carrying 2 copies of the pied gene. Okay, so it is effectively exactly the same as the superform in your incomplete dominant, but in this case, if there was only 1 copy of the gene, there'd be no visual expression. The snake would look normal. You need to have 2 copies of the gene in order for that snake to be a visual pied. So this is 1 half of the pairing. We're going to breed a pied to a normal to see what happens. And I think you already know what happens. In this pairing, doesn't matter which half of this snake you choose, it's going to pass on 1 copy of the gene. 1 copy, 1 copy. 1 copy, 1 copy. So whichever, when you have a visual pied and you pair it to a normal, all the offspring will carry 1 copy of the gene. They're getting a copy from 1 parent, but they're not getting a copy from the other parent. What does this mean in terms of what the snake looks like? Well, with only 1 copy of the gene, remember the blue eyes versus any other eye color, 1 copy of the gene is not enough. It will not show up in the offspring. This guy here looks like a normal. But I know from the pairing that produced this snake, one of the parents was the guy that we've just looked at, the pied snake. So this animal here is what we call het or heterozygous for pied. This animal is carrying 1 copy of the pied gene. Heterozygous and homozygous are very simple terms. Quite simply, if we put the snake away again, het means different, zygous means gene, so it means different genes. So in this particular case, all 4 of the offspring are heterozygous, or hets, 4-pied, which means that they all look like that. They're carrying 1 copy of the gene. The homozygous form would be carrying 2 copies of the gene. Homozygous meaning same. So on both sides of the genetic material we have the same gene. So this is homozygous. And those terms can refer to incomplete dominant genes as well. It doesn't just refer to recessive genes, but we tend to restrict the use of heterozygous or hets just to recessive genes to distinguish them from the incomplete dominant genes. So when you hear the term heterozygous or het, it's usually referring to a recessive gene, and the snake in question is het for that gene. It's only carrying 1 copy of the gene. Okay, so that is what a recessive gene is. Okay, so we've talked about dominant genes where a single copy of the gene, or the heterozygous form, completely changes the visual appearance of the snake, and in fact so powerful is that gene that the superform or homozygous form is actually lethal. Spider to spider produces lethal combinations. So we don't breed dominant genes to each other. We've also looked at incomplete dominant or codominant genes where 1 copy of the gene, or the heterozygous form of the gene, has a visual expression, but 2 copies of the gene is the superform. So we've talked about superforms of codominant genes. We've also talked about recessive, where when the parent carries just 1 copy of the gene, there is no visual expression because it's a recessive gene. You actually need 2 copies of the gene, or the homozygous form of the gene, in order for there to be a visual expression. We've also looked at the fact that these genes occupy their own particular address on the genetic code. And even if you're dealing with the heterozygous form and there is a vacant slot on the other side of the genetic material, a different gene cannot fit into that slot. It can only fit into an address that it has the key for, and most genes have a unique key, so they can only occupy their own individual slots on the genetic code. So 3 different genes would look something like that in the genetic code, and each of these genes acts independently. What happens when we get an allelic combination is where, in actual fact, 2 different genes have the same key and can occupy the same address. An allele is the same address in the genetic code, the same location, and an allelic relationship is where 2 different genes occupy the same address. An allelic relationship produces an outcome which is totally different to what you would expect from looking at the 2 visual parents. Examples of allelic complexes would be yellow belly and gravel, or yellow belly and asphalt, which is the highway or freeway allelic complexes. Another example would be fire and vanilla. They also occupy the same allele. That is an allelic relationship, and the outcome of that is a vanilla cream. Again, a totally different snake from what you would expect if you simply combined fire and vanilla. One of the more common allelic complexes is the blue-eyed leucistic, and there are a number of genes which make up the blue-eyed leucistic complex. There's Mojave, there's Lesser, there's Russo, there's Bamboo, and the various combinations of that. If you have Lesser to Lesser, that will give you a completely white snake with blue eyes. That is also the superform of the lesser gene. However, if we take a copy of Mojave, Mojave has the same key to that address. So we can get a blue-eyed leucistic, which is actually now one copy of lesser, one copy of Mojave. That will produce a blue-eyed leucistic. Now, what does that mean when we look at our genetic computations? Obviously this is a unique relationship and the outcome will be different, but we can still use our genetic calculator. So let's take a look at what would happen if you breed a blue-eyed leucistic male to a normal female. Again, we split the genetic material in half, and because this is an allelic relationship, we now have different copies of the gene in the same address. So if we take this half of the male and breed it to this half of the female, or that half of the female, we transfer one copy of Mojave from the male, no copies from the female, one copy of Mojave from the male, no copy from the female, and then we take the other half of the genetic material, and this time in exactly the same address we have a copy of the lesser gene. So we take this half of the male genetic code and pair it to that half of the female, so we get one copy of lesser, no copies from the female, and we get, breeding it to the other half of the female, again one copy of lesser, no copy from the female. So the outcome will be no normals. Where have we seen this outcome before? We've seen this outcome before in the superform. We looked at pastel, where the male was a super pastel and all his offspring carried one copy of the pastel gene, and that's because each side of the allele carries one copy of the pastel gene, so he has to pass on one copy of the pastel gene. In this relationship, this allelic relationship between Mojave and lesser, or any of the other blue-eyed leucistic complexes, or the highway-freeway complexes, or the vanilla cream complexes, the gene on each side of the allele is actually a different gene. But when you split the material 50-50 and breed it to a normal, you must transfer one copy of one of the genes. So in this particular case, you get half of the babies being lesser and half of the babies being Mojave. You cannot get a normal. There is a gene occupying each of these slots, but it's a different gene. So it acts like a super. This is what we mean by acts like a super. It is actually an allelic relationship between two different genes. Allele is the address. Allelic is two genes occupying the same address. And an acts like a super is exactly what it says. When you split the genetic material in half, you pass on one gene to the offspring. And in this case, it's not the same gene as in the superform, like pastel, where all the babies are pastel. In this case, it's a different gene. So 50% of the babies in this case are lessers, 50% are Mojaves. Also interesting to note is that both the superform and the acts like a super in an allelic relationship cannot reproduce themselves. They get one copy of one gene or one copy of the other. You will never get the offspring containing both the genes. So if you breed a highway or a freeway, the offspring will all be yellow belly and gravel, or yellow belly and asphalt. If you breed the acts like a superform of fire and vanilla, all the babies will be either fire or vanilla, and in the case of blue-eyed leucistic, any one of the blue-eyed leucistic genes, in this case Mojave or lesser. You will not get both in the same animal. In order to reproduce a superform or an acts like a super, the other parent must contain, in the case of an acts like a super, in this case we have Mojave and lesser, the other parent must contain one copy of one of those genes. So if the female is a Mojave, when you take lesser to Mojave, you will get a blue-eyed leucistic. If you take Mojave to Mojave, you will get a blue-eyed leucistic. The other half of the genetic code means that you will just get a Mojave or a lesser. So that's why you need to have a copy in both parents in order to reproduce a superform, or an acts like a superform, an allelic relationship. You will never reproduce it just using one parent bred to a normal. You remember we paired a visual pied to a normal, and we got one copy of the gene in each of the offspring. This is what we refer to, you'll often hear it said, as 100% het for, in this case, pied. Of course, heterozygous means it's carrying one copy, so of course it's 100%. You can't have a different outcome. The offspring are either het for pied or they're not het for pied. But I'm sure everybody's heard the term 66% het or 50% het. How do we get that? Okay, how we get that is, once we've developed our hets, we can breed them back to each other. So now we're going to breed two snakes which are het for pied. And again, using the Punnett square, you can predict what the outcome is going to be, because this is exactly the same as the pastel to pastel pairing. It's exactly the same as the spider to spider pairing. Pair this half to that or to that, so you get one copy or no copies. Pair this to that and you get two copies. This is the superform or the visual recessive gene, so a visual pied, or this half to that gives you one copy. So in this case, where you pair a 100% het for pied to a 100% het for pied, you are going to get one normal, one visual pied, two het for pied. So out of 4 offspring, you've got one visual pied and 3 snakes that look exactly the same. Remove the pied because you know that's a pied. These 3 snakes here now look like normals. How do you know which one is carrying the pied gene and which one isn't? Well, you don't. You don't know. But you do know that 2 out of 3 of the babies will be carrying that gene, statistically, which is 2 out of 3 is 66%. That's where your 66% het for pied comes from. And what you're actually saying is that there is a 66% probability that that offspring is carrying the pied gene. It's either het for pied or it's not. There's a 66% probability that it's het for pied. If you pair a het to a normal, now this is going to be exactly the same combination as when we bred a single copy of pastel to a normal. You get one copy, and you get no copies, no copies. So this time you have 2 out of 4 squares which are het for pied, carrying one copy of the pied gene, 2 squares which are not het for pied. All of these snakes look like normals, so how do you know which one is carrying the pied gene? You don't, but statistically now we're looking at 50% het pied. 50% of the babies will carry the gene, 50% of the babies will not carry the gene. And that's where you get 50% probability of the snake being het for pied. Now in the next episode we're going to talk about working with hets and what that actually means. Why on earth would you ever create hets when all you're going to get is a normal? I think you've seen from the genetic equation here that when you breed a visual recessive, it cannot recreate itself. You only get hets. You need to have one copy of the gene in the other parent in order to get visual pieds. So the gene has to be present in both parents, which means that in that particular pairing, whatever other genes that snake is carrying is the only other genes that can influence the outcome. You cannot introduce new genes into the equation. The only way to get a gene into a recessive project where you do not have those genes present in your visual recessive animals is to create the hets. And you create the hets by breeding a visual to a snake that contains the new gene that you want to introduce into your babies, and that outcome will only produce hets carrying that new gene. And that's the reason why you need to produce hets, and you will hear breeders talking about, I need to produce hets in order to further my projects, and that's the reason why. So in the next episode we're going to use exactly the same method, the same simple 4-square outcome for any particular combination of genes, and we're going to look at multiple gene combinations, and we're going to look at working with recessive genes, what hets can do for you and what the probabilities of those outcomes are. So I hope you found that interesting. I hope it was useful. I hope it explained some of the complex terms which actually are very, very simple concepts. And don't forget to like and subscribe, and we'll see you next time.